Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Haikal Arif bin Abdul Razak, matric number 1170873. So in this video, I will explain to you the stem cell research in industrial biotechnology. Let's get started. In this video, we will go through 5 major contents. The first one is the introduction part, where in the introduction, I will introduce to you the stem cell definition and their classification. The second one is the stem cell in biotechnology, where I will explain to you in details about the usage of stem cell, including the embryonic stem cell ESC and the induced pluripotent stem cell iPSC. Next, I will explain to you the discovery of stem cell in past and present years. In this chapter, I will introduce to you the stem cell in medical application and stem cell therapy. Next, the challenges and obstacles faced by the researchers in studying stem cell research. And then, the last one is the conclusion of this stem cell research. Cell is the simplest form of an organism. The human body comprises of billions over billions of cells in which they function in different ways. For example, the skin cell, the blood cell, the liver cell, and the nervous cell. Each and every cells will function differently according to their specialization. These cells came from the same origin in which the stem cell itself. Stem cell, or also known as immature cells, are cells that not yet specialize into different type of cells. Stem cells are the source of each and every cells in our body. Where are the stem cells located? There are 2 sources of stem cells in human body. The first one is from the embryos, and the second one is the adult cell, or we call as somatic cell. Next, there are 2 key properties or characteristics of stem cell. The first one is the ability to self-renew. This means stem cell can divide and proliferate to make a copy of themselves. Second one is the ability to differentiate, which means they can differentiate or specialize into different types of cells according to their functions in human body. Stem cell is divided into 5 different classifications. The first one is the totipotent, the second one the pluripotent, third one the multipotent, the fourth one is the oligopotent, the fifth one is the unipotent. The totipotent cells are stem cells that are able to divide and specialize into cells of the whole organism, which means they have the highest differentiation potential among the stem cells available. Next, the pluripotent stem cell. Pluripotent stem cells are able to divide and specialize into every germ layers except extra embryonic cells. The example of pluripotent stem cell are ESC and iPSC. The third one is the multipotent stem cell. Multipotent stem cells are able to divide and specialize into the specific cell lineage. For example, the hematopoietic stem cell. Next, the oligopotent stem cell. This type of cells can differentiate and specialize into several types of cells. For example, they can divide into white blood cells but not red blood cells. Last but not least, the unipotent stem cell, in which this type of cells are specific and specialized into certain types of cell only. The embryonic stem cell. Embryonic stem cells originated and derived from blastocyst. Blastocyst is an early stage of fertilization of ovum and sperm. In the blastocyst, there are no organs nor cells. It only contains an inner cell mass, ICM. From the ICM, we can obtain the human embryonic stem cell, in which normally the human embryonic stem cells are obtained from the in vitro fertilization, IVF. The embryonic stem cell will aggregate to form three different germ layers: ectoderm, mesoderm, and endoderm. These three germ layers will then differentiate and specialize into a multipotent stem cell. So, why ESC, embryonic stem cell, are so valuable compared to adult stem cell? This is because embryonic stem cells have the potential to be generated into every cell types found in the human body. Under the right condition and application, ESC can be generated, grown, and expanded into unspecialized and indifferentiated cells. Next, we move to the iPSC, induced pluripotent stem cells. iPSC is the hottest topics discussed in stem cell research. This is because iPSC is a reprogrammed or engineered stem cells to become pluripotent again. The already specialized cells, multipotent, will be induced into pluripotent stem cells to make them behave like ESC. Although iPSC have the same behavior as ESC, they are actually not identical at all. The reason why iPSC is a hot topic in research in stem cell research is because iPSC is a very good alternative to make pluripotent stem cell lineage for specific disease or specific patient. There are several sources of cells in human body that can make iPSC. The first one is the fibroblast, which is the best source for iPSC. The second one is the peripheral blood cells. The third one, keratinocytes. The fourth one, the skin cells. And the last one, pancreatic exocrine cell. So, I will cover 4 past and current stem cell discovery. The first one is the hematopoietic stem cell transplantation. The second one is stem cell in pharmacological testing. The third one is cell-based therapy. And the fourth one and the last one is stem cell in dentistry. So let's begin with the first cell discovery, hematopoietic stem cell transplantation. This therapy or discovery is the most popular stem cell therapy. This is because it has been studied and researched for 50 years. The target cells for HSC are derived from bone marrow, umbilical cord cells, or peripheral blood cells. Hematopoietic stem cell HSC generates all hematopoietic lineage in blood cells such as erythrocytes, leukocytes, and platelets. HSC also solves problems regarding hematopoietic disease such as anemia and leukemia. There are 3 types of procedure in hematopoietic stem cell transplantation. The first one is the autologous, which means the stem cells originated from patient's body. The second one is the allogenic. The stem cells originated from donor. And the third one is the syngeneic. Stem cells originated from identical twin. Next, we move to the stem cells in pharmacological testing. Currently, researchers have found that stem cell can be used as a medium for new drug testing. It can be performed safely as no any live tester are included in the testing. One of the advantages of using stem cells in pharmacological testing is because if any undesired effect occur, the drug formulation can be changed directly so that the drug will develop effective results when tested in human body. However, for the drug to be tested properly, the condition of the drugs tested in stem cells and human body are needed to be equal so that the effects are same. For this issue, the researchers and workers need to understand and fully control the differentiation process in stem cell research. The third discovery is the cell-based therapy. For your information, stem cell can be induced. Like I mentioned before, the induced pluripotent stem cell, iPSC. The usage of iPSC in cell-based therapy are the cells can be grown and generated and then can be transferred to other patients so that the cell will regenerate medicine for the body itself. This is the perfect solution when the transplantable tissue outweigh the supply available in the market. The example disease that can be cured using cell-based therapies are stroke, diabetes, and neurodegenerative disease. Currently, in dentistry research, stem cells is used more than synthetic materials. Enamel, which is one of teeth component, is difficult to regenerate. This is because ameloblastoma is destroyed after differentiated into enamel. So, there are no source for enamel production. Bone marrow stem cell can be differentiated into ameloblastoma, which then will be differentiated into enamel. The first obstacle faced by the researchers are the ethical concern and issues. This is because the isolation of ESC, embryonic stem cell, from embryo could destroy the possibility of the embryo to develop into human. The second obstacle is the immunological tolerance. In stem cells, the immune rejection is the main concern arise among the stem cell research. This is because the immune rejection could possibly affect the donor or the patient in the process of transplantation. The third one is the tumorigenicity. Tumorigenicity means that if the cell is reprogrammed, the potential of the cell to be developed into malignant or tumor cells is high. Next, we move to the future intention by the International Society for Stem Cell Research. There are 5 intentions for stem cells development in the future. The first one is to increase stem cell differentiation efficiency. This is because by increasing the efficiency, more stem cells can be developed in the future. Next, to fully understand the mechanism of stem cell function. This is important in knowing and developing more efficient stem cell research. The third one is to identify and proper isolation of stem cell from human body. This is also important in transplanting stem cell from donor to patient more effectively. The fourth intention is to surpass the immunological rejection faced by patient and donor. This immunological rejection is needed to reduce risk of death and negative effect towards patient and donor. The last intention is for development of self-destruct stem cell. This is because by implementing the self-destruct option, if the stem cells are not suitable for the patient body, it will self-destroy and not harm the health of the patient. Lastly, for the conclusion, stem cell is an important study in science and technologies, specifically in medical application. This is because stem cells provide curing alternative for medical therapies. Stem cells also have many advantages over the synthetic materials used nowadays. However, further work and research need to be done to fully understand the stem cells development in science and technologies. That's all for me. Hope you guys enjoy my video. Stay safe, stay healthy. Assalamualaikum. Bye bye.